Alrighty, welcome, Miss Ray here. Today we're going to be doing our first lesson about genetics. So today you're going to hear a lot of new vocabulary words. We're going to talk about a scientist who started learning about genetics, Gregor Mendel. We'll do some monohybrid crosses. And if you're following along in your book, this does start on page 36 of your blue book if you are following along. So this is page 36. I will be writing it out on this side because some of the stuff I'm going to circle show memory strategies, and I'll be writing out and demonstrating things as we go. Alright, so let's get started. So first we're just going to start off with some review. So our last unit was all about different types of cellular reproduction and how cells make copies to sustain their life. The first type of cellular reproduction that we talked about was asexual reproduction. Asexual reproduction only involves one parent making identical copies of itself. So they're essentially making clones. The main types that we talked about were binary fission and mitosis. Some pros of this type of reproduction are all organisms look the same. There's no need to take the time to go out and find a mate. It's fairly fast and efficient, but all the organisms are identical. The cons is that there's no genetic diversity. All the organisms are the same. And in our world, being diverse, having differences, are very beneficial for long-term survival. And the type of reproduction that gives that genetic difference is sexual reproduction. So sexual reproduction involves two parents, and it leads to genetic diversity. The process that makes these sex cells, the sex cells will eventually become together to make a baby. The process that does this is called meiosis. Meiosis is a process that makes haploid gametes. Remember, haploid means contains half of the genetic material. Gametes are our cells that are going to come together to make a baby. In males, the gamete is the sperm, and in females, a gamete is the ova or the eggs. And meiosis is the process that creates these cells. So the way that this works is we have the mother contributing 23 chromosomes in its egg cell. That will combine with the sperm, which carries 23 chromosomes from the father, and in the end we end up with a diploid zygote. A diploid zygote means it has a full set of chromosomes, getting one set from mom and one set from dad. The zygote here will have 46 total chromosomes, which is the normal number of chromosomes for a human. Therefore, it is diploid. 2 copies of each chromosome. These 46 chromosomes are arranged in homologous pairs. They are arranged in homologous pairs. They get one set from mom and one set from dad. So you get one copy of every chromosome from mom and one copy of every chromosome from dad, and then that will give you your homologous pairs of chromosomes. These homologous chromosomes are the same size, shape, and code for similar genetic information. So you get one copy of eye color from mom, one copy from dad, so on and so forth for all of your different genes. And the combination of genes that you inherit from your mother and your father in the homologous chromosomes, the combination that you get, this is going to determine your traits, what you look like, who you are. And that leads us very nicely into our discussion about genetics. When we start discussing genetics, we have to start with the father of genetics. But first, let's go ahead and have a question. So question number 1 is, where does a person get their homologous chromosomes? Where does a person get their homologous chromosomes? All right, so continuing on, the father of genetics. The father of genetics, his name is Gregor Mendel. So Gregor Mendel is the father of genetics, and he did all of his work by studying garden peas. Now, you may be asking, of all the things in the world that he could study, why would he choose to study a garden pea? And the reason behind that is because garden peas have traits that are very easy to see and study. The traits of garden peas are very easy to see. Some examples are flower color, seed color, and seed shape. Now, he also looked at some other traits, like the height of the plant as well. And we'll talk about some of those traits that he looked at a little later. But all these traits, they're either or traits. The flowers are either purple or white. The seeds are either yellow or green. The seed shape is either round or wrinkled. There's no mixing of traits. It was an either or situation, making these traits very easy to study and examine. Not only that, but he had strict control of which plants mated together. So he knew the genetics of the parents, and then he could analyze the genetics of the offspring, which gave him the optimum amount of data and control in his experiments. Pea plants are also, since they are plants, they have many generations in a very short period of time, and you get many offspring for every single cross that you do, which means he could look at lots of offspring and lots of generations in a very short period of time, giving him the ability to have lots of data and to study trends over generations over time, which was very helpful. So let's go ahead and look at what he did. But first, question number 2. Number 2, why did Mendel study pea plants? Why did Mendel study pea plants? So in his experiments, the first thing he did is he cross-pollinated true breeding parents. True breeding is another way for saying purebred, or 100% for whatever trait you were talking about. So what he did in this case is he was looking at the flower color. He crossed a true breeding pure white flower with a true breeding pure purple flower and looked at their offspring. And he observed the traits that the offspring had. And that generation is going to be called the first generation, abbreviated with F1. From there, he took the F1 generation, or the purple flowers, all of the offspring from the true breeding white and the true breeding purple, all of their offspring were purple. So he took it one step farther and went ahead and self-pollinated these purple flowers, which essentially means they mated with themselves. So an F1 mated with an F1. And from there, he looked at the second generation, which is called F2. So when I write that out here at the bottom, what he did, the very first generation, the parent generation, P stands for parent. In the parent generation, he took a pure white flower, mated it with a pure purple flower, looked at the F1 generation, the first generation of offspring, and those offspring were all purple. Then what he did is self-pollinated these purple flowers to get his second generation. And he noticed something interesting happened here. When he self-pollinated the F1 generation to get the F2 generation, the second generation of offspring were 75% purple and 25% white. And this was curious to him because he hadn't seen the white flowers since the very first parent generation. The color white, this trait, skipped a generation, and he was wondering, how does this happen? How can a trait skip a generation? How exactly does this work? And that led him to some more of his studies, which we will talk about here in a minute. So what he found, let me go ahead and switch my page. What he found is that for each characteristic or trait that an organism has, they inherit 2 alleles. One of those alleles comes from mom and one comes from dad. Now, the word allele just are versions of a gene. So in humans, when we look in humans, they all have hair. But they can have different colors of hair. You could have brown hair or black hair or blonde hair or red hair. And sometimes they even, other colors other than that, a little bit of mix. Brown hair can be dark or light. So there are many different versions of genes, and those are called alleles. So he knew that these genes, these alleles, came in many different versions. So the word allele just means a version of a gene. And what he noticed from there is that some traits can mask others. Question number 3. What is an allele? So question number 3, what is an allele? So he found some traits can mask others. When we're looking in flowers, the flowers are either purple or they are white. There's no mixing, they're either purple or white. And we know that there are different versions of genes or alleles. And what he came to conclude is that some of these traits, if you have a gene for one of those flower colors, it will cover up the other. In this case, in the flowers, the purple color hides the white. So if one of your parents gives you an allele for purple flowers and the other parent gives you an allele for white flowers, you have purple flowers. If you get one copy for having purple flowers, your flowers are purple. It doesn't even matter what your other allele is. It covers it up. It's like the other one isn't even there. And what we call this allele that is expressed, the one that covers up another, that is called the dominant allele. The dominant allele is the one that's expressed. It's the one that masks others. It covers it up. Because it is the one that is dominant, covers other things up, it's going to be represented using a capital letter. We use an uppercase or a large capital letter to represent the dominant allele. Now the other allele that is easily covered up, the one that's masked, like the white color that's easily covered up, that is called the recessive allele. So the recessive allele is the one that is easily covered up, it is masked, and we're going to represent that with a lowercase letter. We use the smaller letter to represent the trait that is often covered up or masked. So here's the question. This is question 4. In dogs, if black fur is dominant to brown fur, which describes the brown fur allele? And select all that apply. So far we've talked about 2 different things. We've talked about the way that an organism looks, in this case, the color flowers, purple or white, but we've also talked about the genes that you get from your parents, the homologous chromosomes that you inherit. And there are going to be words for those. In an organism, the physical appearance is called the phenotype. The way that I remember this, pH for physical appearance. pH, phenotype, physical appearance. This is what an organism looks like. You look at it, you see it. That is the phenotype. On the other hand, we have the genes that the organism inherits, and that is called the genotype. So the genotype, I remember geno for genotype, genes. These are the genes, or the genetic makeup of the organism. So when we are looking at flower color in pea plants, the phenotype for color is going to be, well, are they purple flowers or are they white flowers? The genotype, on the other hand, is the combination of letters that you inherit from your parents. So you can either inherit 2 dominants, both mom and dad give you the dominant allele for purple. One parent can give you the allele for purple and the other can give you the allele for white, or both of your parents could give you the allele for white flowers. These genes that you inherit make up your genotype, the genes that you have. Question 5. Select all that apply from the list. Which of the following are phenotypes? So what we can do is we can make crosses, and that's what Mendel did. Crossing means mating of flowers. And we're going to use these letters to represent various crosses. So essentially, going back to the work that Mendel did with his pea plants, we're going to use the letter P in this case. You could use any letter you want as long as they're the same. So as long as you used the same letter, you could use an A or a B or an R. It doesn't matter. You can use any letter that you want. In this case, they chose P for purple. But again, it doesn't matter. Any letter is fine. I typically recommend choosing a letter that is different, capital and lowercase, like an A or an R, because that way you're not going to confuse yourself whether you wrote a capital P or a lowercase p. So in this case, I'm going to make my capital P very big and my lowercase p very small. So the dominant allele in this case is going to be represented with a capital letter, so a capital P. And the capital P, remember, dominant alleles cover up other ones. This was the purple. Capital P stands for purple. The recessive allele, the one that's easily covered up, we're going to represent with a lowercase letter, and in this case, the color that was easily covered up is white. And when Mendel was doing his work, he first crossed a true breeding pure purple flower, which would be capital P, with a pure white flower, which would be lowercase p. Remember, all parents, all organisms, have 2 copies of every trait. They only give 1 to their children. So while parents have 2 letters representing 2 alleles that they received from their parents, they only pass 1 to their child. So Mendel, in his work, remember, he crossed a pure purple and a pure white. He looked at the first generation. They were all purple. Then he self-pollinated the purples to look at the F2 generation. If we were to write that out using our letters, the parent generation were pure purple crossed with a pure white. And then when we look at the F1 generation, we can figure out what letters the F1 generation has based on what the parents have. So parent number 1 here, this is parent 1, this will be parent 2. Parent number 1 passes their child only 1 of their P's. But since this parent only has capital P's, their child gets a capital P. They only get 1, but it is capital. The same for parent 2. Parent 2 here only has little p's to pass down, so their child has to get a little p. Therefore, when we are looking at the F1 generation, the offspring of that parent generation, we know that the F1 generation all were big P, little p, based on the genetics of the parents. We can figure out that the F1 generation was big P, little p. Then what Mendel did is he cross-pollinated, or self-pollinated, these F1 purple flowers here, and that's how he got his F2 generation. Now, the way that we are going to represent our work when we're looking at genetic traits is in a Punnett square. A Punnett square is essentially a small table. It can be various sizes depending on how many traits we're looking at, but it helps us predict the chance of the offspring having a certain genotype or a certain phenotype. So when Mendel self-pollinated his purple flowers, parent 1 was big P, little p, and parent 2 was also big P, little p. So these are his first generation flowers that were self-pollinated. This work that we're about to do is going to give us the F2 generation information. So when we're working with Punnett squares, I'm going to show you how we fill it out, but don't worry, we're going to have more practice here in a few minutes. So I like to always label my parents. So I'm going to pull out and use some different colors here. So this is going to be my parent 1. This is going to be my parent 2. So I'm just labeling my parents there. Now, parent 1 only passes 1 of its letters to its child. It can give its offspring either a big P or a little p. Because each parent only passes down 1 letter to their child, we separate them when we're filling it out on the square. Also notice at the top, this says father genes across the top and mother's genes along the side. it doesn't matter which parent goes where as long as one parent's along the top and the other is along the side, and you separate the letters when you fill it out. So if you confuse mother and father, it doesn't matter. A lot of the times, you're not even going to know which one's the mother and which one's the father. It's just going to say parent 1, parent 2. And that's totally fine. Parent 2 has a big P and a small p. It can either give its child a big P or a small p, but because it only gives the child one p, we separate them here on the square. Now we're going to figure out our offspring. Each box here represents a possible combination for each offspring. So we're going to have 4 different possibilities of what the offspring could look phenotypically or genotypically. We will have phenotype and genotype represented here. So the way that we fill this out is just like a multiplication chart. This big P will go in this box. This small p, or that's another big P, my bad, will also go here. When we're filling out box number 2, big P moves over. This small p comes down. And this is just like a multiplication chart. Big P will come down. When you have this box here, you could write the small p in front, but typically scientists are going to always write the capital letter first. Our last box, little p, little p. So essentially they combine like this. This box comes from these parents. This box comes from them. This box, this box. And you fill out your square. Now we're going to look and figure out the genotype and phenotypes of the offspring. Again, my offspring are what's represented in the inside boxes. I like to do genotype first. One, it's written first, but also because the genotype are the letters, and the letters are what we already have, so you don't have to do any work. They're already there for us. All right, so genotype percent of the offspring. All right, let's see. How many big P's did we write inside the squares? Just this one. 1 out of 4 was big P. 1 out of 4 is 25%. It's convenient that we have 4 boxes here because you can think of each like a quarter. 4 quarters make $1. 25, 25, 25, 25. So this is 25% or big P. All right. Next, how many big P, little p's do we have? Or offspring 2 and 3, those are both big P, little p. So there are 2 out of the total 4. 2 out of 4 is 50%. 50% chance that the offspring could be big P, little p. Next, how many little p's? Just offspring 4. 1 out of 4 is 25%. There's 25% chance that these offspring are going to be little p. Now we need to look at the phenotype. Remember, phenotype is what you look like. Phenotype is what you look like. There are 3 possibilities for the genotype combinations, but there's only 2 possibilities for what they could look like. Remember, big P was purple, little p was white. They could either have big P. Because big P is dominant, it's purple. They could have big P, little p. Big P, little p, it has a big P. It doesn't matter what this other one is, it is purple. If it has a big P, it is purple automatically. The only way that the flower can be white if there are 2 little p's. So big P equals purple. Big P, little p is purple again. Big P, little p, purple. Little p is white. So figuring this out, how many purples? 3 of the 4 are purple. That's 75%. There's a 75% chance these offspring are going to have purple flowers. Only 1 out of the 4 has white flowers, so there's a 25% chance that the offspring will have white flowers. Now, if you notice here, like I wrote on the side, there's various combinations that these organisms can have. We're going to have words for that. But first we have a question, question 6. Why did the trait for white flowers skip a generation? So like I was saying, there's many combinations of letters that these organisms can have. Organisms are considered homozygous. Homo means same. Zygous mean alleles. They have 2 of the same alleles. So an organism that is big P, 2 of the same letters. Or little p, both 2 of the same letters. They're both homozygous. An organism can also be heterozygous. The prefix hetero means different. Zygous means alleles. They have different alleles, meaning they have a capital and a lowercase. So heterozygous organisms have 1 of each letter. A homozygous organism has 2 of the same letters, in the sense that they are capital or lowercase. So essentially what this is saying is that 2 organisms, they can have the same phenotype. They can look the same, but they can have different genes. So 2 organisms can look the same, but have different genes. Okay, question 7. Choose all of the homozygous allele combinations. Now, because 2 organisms can look the same but have different genes, we might have a problem. If I had a purple flower, I wouldn't know its genes. I can look at that flower and be like, that's a purple flower. I could know its phenotype, but not know its genotype and not know its genes. But I could figure it out. So I could figure it out if I just perform a cross. So if an organism had big P, little p, it would be purple, and it would be considered homozygous dominant. Homozygous, 2 of the same, dominant, capital. But my purple flower could also be big P, little p, and it could be heterozygous, meaning that it has a big P and a little p, 2 different alleles. I don't know what genes it has until I perform a test cross. A test cross will help me figure out the genotype of my flower. And the way that I do this is by breeding this unknown flower with one that is homozygous recessive. In this case, homozygous recessive is small p, 2 of the same, small letters. And in this case, little p, that flower would be white. So I take my unknown purple flower and cross it with a white flower. And the reason this works is we're going to look at this in the format of a Punnett square. The reason this works is because when I cross it with a white flower or a homozygous recessive flower, I can look at the offspring and see what they are, and that'll help me determine the genotype of the parent. So my purple flower, it could be big P, or it could be big P, little p. In both cases, I'm going to cross this unknown flower with a little p, a homozygous recessive white flower. From there, I'm going to fill in my Punnett square. But because this flower here only has capital letters to give, if my purple flower is big P, all of the offspring will get a big P, meaning all of these offspring are going to be purple. 100% of these are purple because they all got a big P. However, if my purple flower was big P, once I fill this in, I'll see half of the offspring will get a big P and be purple. So half will get a big P and be purple, but the other half is going to get a little p and B, white. So if I do this test cross, if any of the flowers come back white, my organism was big P, little p. If all of the flowers came back purple, then my unknown was big P, big P. And from this, Mendel created his laws of heredity. The first law of heredity is the law of segregation. And essentially what this law says is that during meiosis, pairs of alleles separate or segregate. All that means when we're talking about what we've talked about so far is parents have two letters and they only give one of those to their child. So they separate. That's why we separate them when we're writing them on the Punnett square. That's why I separated them here. Because the parent is only going to pass down one of the letters to its child. So that is the law of segregation. A parent's two alleles will separate and the child only gets one of each. Question 8. Which is true about the law of segregation? Now, when we're looking only at one trait at a time, we are performing monohybrid crosses. Monohybrid crosses, the prefix mono means one. We're only looking at one trait at a time. And we did one earlier, but we're going to go through another. And I'm going to show you some tips on how to walk through these problems. First off, let's read our problem. Number 1. Tall is dominant to short in pea plants. Cross a heterozygous tall plant with a short plant. What are the phenotype and genotype ratios? So step number 1 is to figure out a key. That means we're going to figure out what's dominant, what's recessive, and give it a letter. Tall is dominant. Okay, tall is dominant. That means tall is going to be represented with a capital letter. Let's use T. Big T for tall. Tall is dominant to short. That means short is recessive. Recessive things are represented with a lowercase letter. So tall, this one is the dominant one. Short is recessive. Meaning if an organism has a capital T, it's tall, automatically. Doesn't matter what the other one is. All right, step number 1, check. Step number 2, figure out your parents. All right, now we're going to be looking for cross a whatever, that's going to tell us our parents. Cross a heterozygous tall plant with a short plant. That tells me one of my parents is heterozygous tall, the other one is short. Heterozygous means different. Parent number 1 is going to have one of each letter, a capital T and a lowercase t. Remember, parents have two letters. They only give one to their child, but they have two. Parent number 2 is short. That means they have a little t, because little t is short. But because all parents have two letters and this parent is short, they have to have two small t's. Number 2 done. Now we draw out our Punnett square. Make sure you have enough room to work around it. Remember, as long as we fill this out correctly with one parent on the top, one parent on the side, we're not going to mess it up. I'm going to put parent number 1 along the top. Parent number 1 can either give a big T or a small t. Parent number 2 can either give a small t or a small t. And then I fill this table in just like a multiplication chart. Filled it in. Yep. Now we have to answer our question. Our question asks for phenotype and genotype ratios. I like doing genotype first just because it's already written there. So let's do genotype. My genotype possibilities: 2 capitals, a capital and a lowercase, or 2 lowercases. Alrighty. Now, here we have 4 possible offspring combinations. 1, 2, 3, 4. How many are big T, big T? 0. 0 out of 4, that gives me 0%. Now we have big T, little t. 1, 2. There's 2 out of the possible 4 are big T, little t. That is 50%. 50% chance they're going to be big T, little t. Now we do little t, little t. 1, 2. 2 out of 4, that's 50%. 50% chance they're going to be big T, little t. I mean, little t, little t. Now we just have to do our phenotypes. Phenotype is what they look like. These ones can either be tall or short. Remember, big T means tall. Big T, this one is tall. That one is short because there's no big T. This one has a big T, so it's tall. This one, short, because there's no big T. In this case, 2 out of my 4 are tall. That's 50% chance they're going to be tall. 2 out of 4 are short, so that's another 50% chance that these offspring are going to be short. All right, so question 9. What was the dominant allele in this problem? Question 10. What does heterozygous mean? Question 11. Why are the letters of the parents separated when we put them on a Punnett square? Question 12. What do the letters in the boxes represent inside of these boxes? Now, our last few questions are going to ask you to go through your own problem. Specifically, I chose problem number 2 on the back. The problem reads, round seeds are dominant to wrinkled seeds in pea plants. Cross a homozygous round with a heterozygous round. What are the genotypes and phenotypes? Question 13 for that problem. What is the dominant trait in this problem? Question 14. What are the alleles for the parent that is homozygous round? Question 15. What is the chance that the offspring from this cross will have wrinkled seeds? Okay, guys, this stuff takes a lot of practice. It's a lot of words and it takes a lot of practice. Please see your teacher during office hours if you have questions.